Good morning, ma'am.
Good morning. This is Dr. Shampadev. Okay. I will be starting with genetics and principles of variations, as you call these. Can you see these absolutely properly? Is
Yes,
the
ma'am.
writing visible?
Yes, ma'am. It's visible.
Because, for genetics, we have to understand these writings very well. So if there is a problem, just tell me, okay? Just inform me. All right. So we will start now. You all know the meaning of genetics, right? Genetics is the study of heredity, right? So what is the meaning of heredity? Heredity is something which is transferred, or rather I should say transmitted from one generation, from parents to the offsprings, okay, throughout generations, through many generations. This is what we call as genetics, okay. So the geneticists are the ones who study the type of propagation of such kind of hereditary characters to their offsprings, their structures and all these things. Okay, so this is what is actually dealt in with genetics. When we come to genetics, you understand that we are using these terms heredity. What is the meaning of heredity? You all know, but since we are starting it for the first time in this time, so I think we should give a brief discussion of all these things. Okay, so heredity simply means the transfer of exactly, or I should not use this term exactly because we are talking about genetics. Okay, more or less the same kind of trait or characters which are present in parents, it should be passed to the next generation almost, exactly as it is present in the parents. So why I'm using this term almost? Because never, when you are talking about sexually reproducing animals or plants or microbes, anything that is reproducing sexually, never will be the offspring is exactly, ditto, as a Xerox copy of their parents. Hmm? That occurs because of the transmission of characters from both the parents, which we now know. Okay, now it is known that we uh, have got the genes from both the parents transmitted to their generations via the egg and the spermatozoa. Okay, in non-sexually reproducing animals, what happens is that either of the parents, we usually call it as a mother cell, so that is going to divide exactly into halves, and subsequent halves will go from one generation to the other. So there will be no mixing of, as you are talking about, male and female characteristics in these. So what happens is these, in the next generation, the individuals will be having only a very little variation, what you call as say maybe about 1 or 2% of variations you can see from the parents, okay, which is passed on to the offsprings. But if I'm using this term passed on from the parents, that is not right, because in this case of asexual or non-sexual reproduction, as we are talking about, they usually have these changes, or I will use the term variation. Now I'm going to use this term variation. Can you see that? Visible?
Yes, ma'am.
Okay. So this variation is what we are actually going to use now, which simply means the difference between the structures or appearance of the parents from the, or rather, the offspring from the parental generation. So these variation that we see in asexually reproducing animals mainly come from the environment. Okay, so this is a kind of environmental change that we see. So that is hardly representable. Mostly they will be exactly similar to the parents. So we call them as clones. That means the characters that they inherit from their parents will be exactly like what is present in the parents. But when we come to the sexually reproducing animals, the case is absolutely different. As you know, in sexually reproducing animals, two different individuals will be involved for the reproduction. What happens is, in most of the time, we see, suppose we are taking a male parent here, so like this, and a female parent like this, the offspring that we are going to get here will be mostly an intermediate, okay, of the two. But this intermediate doesn't mean that they are going to inherit half characters of both these, because many things actually decide what will be the characters of the offsprings here. The variation seen here is tremendous, actually. More than 90% of variations can be seen in different individuals that are born from the same parents. It is because of various reasons. Okay. Even though we're talking about the intermediate, this term is exactly not right, as I have already told you that these things are actually dependent on many factors. All right. So variation is something what we are telling, or rather we can call something which is different from the parents, which are not observed in the parents exactly in the same which is present in the offspring. I hope that is clear to you. All right. Now, the variations that we find in case of sexually reproducing animals, we will discuss first, because it is almost non-persistent in case of the non-sexually or asexually reproducing animals. So sexually reproducing animals, variations are different kinds. The first one is what we call as somatic variation. Okay, the second one is germinal variation. So this is something which is very important for genetics. As you all know, somatic simply means the body cells which are not a part of the reproductive tissues of the males and the females. So any kind of variation that is obtained here by the parents, by either of the parents, is absolutely unlikely to pass on to the next generation. But of course, it actually forms a basis of variation. Okay, so how do we get the somatic variation? This is usually obtained by, this is what we're calling as acquired variation, which may simply happen due to some kind of mutation, okay, small or large, that we will come to later. Next will be the environmental changes. Here also environment plays a role, environmental variation. And the third one is actually constant effort or conscious effort by the individuals, by which we get some changes in these. Okay, conscious efforts. What is the meaning of these conscious efforts? You can understand these if I tell you that if you want to grow stronger, we simply have to do some kind of exercises, run, or different kinds of exercises done. This is just one example of conscious efforts by which changes can be brought to only the somatic cells of the body. But in case these changes are brought about by some kind of mutations which is occurring in the reproductive cells, of course, this will be passed from the parents into the offsprings. Okay? So I hope you're understanding what we are talking about. All right. So we have come to terms like heredity. You will understand the meaning of heredity is simply transmission of traits or characters. Usually, we use these terms, simultaneously, traits or characters, even though traits is something different from characters, which we are going to discuss later. Okay, so transmission of traits or characters from one generation to the other is what we are calling as heredity. We have discussed what is genetics, and we have discussed also about the variation, but only about the somatic variation. I will start with it a little later, okay? About these germinal variation, but before that, we have to speak about something else. You all know that Gregor Johann Mendel is known as the father of genetics. Okay, but it is not right that no studies on the heredity was conducted before his time. Okay, continuous efforts were being made to understand how is it that almost the same kind of characters are observed from one individual to the other. Like, as you have already done, these mango trees will always produce mangoes, oranges will produce oranges, and so on. Okay, so how is this passing? So there have been different theories put forward for these. So we will call this as pre-Mendelian assumptions or theories, whatever you want to call this. Many people actually spoke about these transmission. They tried to explain these in various ways. Many different types of scientists were involved in this. We are not calling them as geneticists, even though we should, because they wanted to find out how this transmission of characters from one generation to the other occurs. But at that time, you have to understand, because Gregor Johann Mendel has actually published his papers in 1856. So that was long before the meiosis or anything about nucleus of the cell or mitosis or any such thing was known. He was actually much ahead of his time. You can understand now, you know about mutation, and you can know about this meiosis, how actually during meiosis the recombination of genes occur, which we are going to talk about when we are talking about this germinal transmission or germinal heredity. So recombination occurs during these. He had no idea of these things, but he had proposed about these mixing of these characters now, which we are calling as recombination. And he was the one to say that these combination of characters from the parents actually bring about the changes, or rather the differences between the parents and the offsprings. Okay. But before him, the theory or pre-Mendelian assumption, what you can call, they used to call this as blending. Okay, blending, you understand, blending is simply mixing of two different characters. So this was known as blending. So what happened in this blending or blending theory of inheritance simply says that two parents, okay, when they reproduce, when two sexually reproducing parents are taken, the offsprings that we get from them will simply inherit half the characters from one parent and half the characters from the other parent, thereby, mingling or blending, okay, of the characters of the two parents, producing an intermediate between the two. And this is what we are talking about is not right always, even though I'm using this term, but this is what was in vogue. They used to say that the characters of the two parents will mingle or blend together, producing intermediate. Suppose, for example, we are talking about two cats. One was black furred, and the other we take as white. So, expected result will be gray because that is the intermediate between the black and the white. Okay. Yes, in some cases that uh, does happen, as you have seen in this, what was that? Incomplete dominance, right? But this is not always the case. This is just, in some cases, you are going to get a blending or the intermediate features. So this actually did not, even in that time, that was around, you can say, late 19th century or early 19th century, whatever you prefer. In 1800s, this was the idea that both the parents actually give equally to the formation of the characters in the offsprings. Darwin, as you know, had given his theory later, pangenesis and all these things. What did he say? He had got little ideas from the blending theory for his pangenesis. We are not talking about evolution here. We will simply talk about pangenesis, which he had actually spoken of regarding these genetics. He said you might have heard about pangenesis. It simply says all the organs of both the parents will form very small structures, which we call as gametes. Each one of the organs, okay, they will form gametes, which will be actually accumulated in the male gamete, and that will be transferred from one generation to the other, bringing about the changes that we find in the offsprings. Okay, but you know that is not right. This is not what happens. Not all the organs produce gametes, and this goes to the reproductive organs to pass on to the next generation. So in late 1800s, Weizmann, which is also very well known for his germplasm theory, August Weizmann, had actually proposed that only he what he said was, in every individual there are two different kind of plasms. Okay, he used this term, plasms. So he said there are somatoplasms in individuals, and the other one is known as germ plasm. Okay, so somatoplasm is actually which is not taking part in the reproduction of the organisms at all, and these can undergo changes, as we have already mentioned, like environmental changes or conscious effort or any other kind of mutations and all these things. Unless these changes are brought about in the germplasm, this will not be transmitted to the next generation. The same thing we can say in other words, the offsprings will not inherit any change that is brought about in the somatoplasm of the organisms. Okay, but any changes that is brought about in the germplasm of the individuals, that will be inherited by the offsprings. Yes, he was not able to explain it very well as Mendel had explained, but you can say that this is something which is very important to understand the process of genetics or the theory of inheritance also. Okay, now we have to know of some terms, but you all are aware of these. But I think when it is better for us to start with the Mendel's theory and all these things, how he had started his experiment, how did he perform all these things, and then if we come to these terms, it will become easier for us to understand. So let us start with it. As I have already told you, Gregor Johann Mendel was his name, and you are aware that he was an Austrian monk, and he came from a farmer family actually, and they had apple orchards. He had known about these cross breeding between apple of various kinds to bring about a very good one or a better tasting one, like that, since his childhood. And that actually intrigued him. He wanted to understand how is it possible that suppose you take a sweet apple, a small one. This is sweet in taste, and you have a very large apple which is not so sweet in taste. But you can understand the flesh that is present in these is much more than we have in these. So it has been actually, it is more than 1,000 years or that breeding experiments have been done. Okay, without understanding the underlying process of how these changes are brought about in the offspring. But of course, that has intrigued mankind, and many such kind of experiments have been done without proper understanding of these. And when these two were actually crossed together, we will get an intermediate which will be larger than the first parent, yes, smaller than the second one, but it will also be sweeter than this one. So, since he knew that the characters from both the parents are actually coming to the offspring, he wanted to understand these. And when he was the priest in the church, he had a garden. Okay, so he started working with, uh, you all know these, garden pea, we call them. The scientific name is Pisum sativum. So why did he choose this Pisum sativum for his experimental material? There are many characters which you find in garden pea, and for any experiments, you have to have any such kind of experiments. I mean, for what you want to see in heredity, there has to be some characters which are very well pronounced, okay, in the parents, that so that you can also observe them in the offsprings. So that was actually very, very pronounced in garden peas. There are certain characters which you can see in them very well, and which are transmitted from generation to generation also. There are many other characters which the garden pea has. They are annual plants, these, they complete their life cycle just in one season. So one seasonal plant. Next is very well distinguished characters. Number 3, they are true breeding. Now there, here we have to understand what is true breeding. I will explain you later. Let me just finish these. True breeding, they are naturally self-pollinating, but they can be easily cross-pollinated as well. Individuals that are produced are small, and You can count the numbers and all these things. So in genetic studies, these are very important. So there are many such characters which are usually found in the garden P, for which Mendel had chosen these characters. Okay, I mean these plant as his experimental uh, thing. Now we are going to see the meaning of true breeding. But before that, we will come to, if you remember, we had spoken about traits and characters. And I had told you, even though most of the time we use it for the same meaning, it is not exactly true. So let us come to what is the meaning of character. This is an expression of the organisms which you find in them. Okay, let us not go into the internal characters. We will only speak about the structures or appearance of the individuals which we can see with our eyes. We are only calling these as characters in this case. Okay, say if you're talking about the garden P, there are certain features which are present in them, and each feature is what we are calling as a character in this case. Say, for example, color of the plant. This is a feature present in the plant. We are calling this as a character. Then say height of the plant is another, height of plant. Then you can take any other, you can take the color of the seed coat. You can take texture of the seed coat. Okay. There are many such kind of characters which you see in the individual pea plant. But if you come to this color of the plant, color of the flower, I should have written this here, color of the flower. When we come to this flower color, you will have many variations in the color, right? You have seen red colored flower in case of peas, and you have also seen white colored flowers, okay? You can say these are two contrasting features of the same character. What is character? We are talking about color of the flower as the character. But when we look at the color, we can see two different variations in these. Okay, there are many other variations, but we will just stick to these now for the time being so that we understand this well. And then, of course, we will understand the rest better. So I'm talking about only two opposite expressions. Okay, one is red and the other one is white. Very well differentiated from each other. Observable changes. Now, each of these, these variations that are present in the character of any individual is what we are calling as a trait. Is that clear to you? Even though, first of all, we said character or trait that is transmitted from one generation to the other is something what we are looking for. But actually, these two are slightly different from each other. Character is something, which is present in an individual, and trait is something which simply differs from one individual to the other. The same character, but they have got, you can say, different kind of variations, contrasting. Okay, feature that we find among the characters is something which we can call as a trait. I hope that is clear to you. If there is any problem, you will please tell me. Okay, unmute yourselves and you will tell me, or you can also do this. You can write me. Okay, you can send me your messages later to clarify your problems. If you don't understand anything, my writing or my explanation or anything, any terms that I have spoken about now is not clear to you and you are unable to tell me now, please let me know later. Okay, I will try to clarify these through WhatsApp messages. Okay, I hope that is clear to you. What we are calling as a character is something, a particular thing, character, which we find in individuals, and trait is simply the variations of the same character that is observed in the individual. I hope that is clear to you. We will come to height then. What will be the traits of the height? we will only consider, even now I'm telling, here we are just going to consider the contrasting features that we find in them. We are not going into these intermediates, which will be present in these. So if you're talking about height, we can take two contrasting features. Some plants will be very tall and some plants will be dwarf. Okay, like that. If you're talking about seed coat color, usually two different colors will be present here, yellow and green. If you're talking about the texture of the seed, again, then we will take only two variations which are contrasting. So first one will be round and the other one is wrinkled. I hope that is clear to you, the difference between the character and the trait. Now, we will start with the experiments of Mendel. How did he start with his experiments? And there are some terms regarding these. So as we finish each of these, you will understand these terms as well. Uh, Mendel had actually started his experiments with these pea plants, and he had chosen 7 characters. You're aware of these. You know these well enough. 4 we have discussed. Okay, 3 are there. 3 more are there. So I would like you to tell me these, but I know this is not possible in this Google Meet. That is why I'm asking you to converse. Okay, through WhatsApp. That will be better. I mean, instead of my giving you a lecture just like that, and you are just trying to take that in. And since this is our first day, we have started with genetics. It may not be very clear to you. So that is why I'm telling you again and again, if there is any problem, do contact me. Okay, so we will start with these experiments of Mendel. The most well-known, he had actually got the same kind of results for all these experiments he had conducted with the 7 characters he had chosen in the pea plant. But the renowned one, the most renowned one, is the one that was done with the character height of the plant. So we will start with that. And then, when we go through these experiments and the results that he had obtained, we are going to also understand the terms that is associated with it. So first, we are going to choose these height of plant. And here again, as we have said, we're just going to choose the contrasting features so that it is easier for us to understand how these are transmitted from one generation to the other. So in height of the plant, we will take 2 traits. Now we are using this term. One is tall and the other one is dwarf. So what we have to do is, what he did rather, he chose 2 plants. The one of the plant was tall and the other plant was dwarf. Okay, so he chose these are his terms. Okay, some terms were given later, but quite a lot of the terms were given by chosen by himself. All right, so what he chose is, he called them as P plants. P simply comes from parental. Okay, so parents, P, parents. These are the first parents. So this will be known as P1, the first parent, and this will be known as P2, the second parent. All right. So as we have already said that the pea plants are self-replicating, but what we want to do now is we want to stop self-pollinating and we want to do cross-pollination rather. So what is done? They are going to, this is what Mendel had done. Dwarf plant was actually chosen as the female parent, first of all, but always this is not the case. I will just tell you for our understanding better. I'm just taking this as the female parent now. Okay, so what you have to do, since you know that this plant also will have the male parts in it, so you'll have to just remove these male parts. Okay, anthers and all these things will be removed from this. You have a term for this, right? This is what you call as emasculation. You simply remove the male part from it. After emasculation, what you have to do is That means is you don't allow self-pollination, that's it. Okay, so from the tall plant, you are just going to dust the flowers in this dwarf plant with the pollen from tall plant. So that is what you are calling as cross-pollination. Now, these being done, these dwarf plants will have to be covered up. That can be done by polythene bags, okay? And I have also seen, people just turning it like this, bending it so that pollination is not possible. I mean, then this problem of having plastic bags and all these things does not arise at all, okay? But that is done practically. So what happens is this, for our convenience, we will just use this term. So covering this is what you call as bagging. You simply cover it so that no other, some stray pollen grains does not come here. Okay, so that is bagging. And you can also note from which plant you have used these pollens on which date and all these things. So you keep these records, okay? You keep a track of these experiment. So that is what we call as tagging. We write name from the source from where the pollen was taken, on which day, and so on. This is what we call as tagging. So these are terms which are utilized in this process. Now, what we have to do is just wait for these plants to reproduce. Okay. What Mendel had actually done, even though for our convenience, we are calling this as a female parent and this as a male parent, but initially what he did was he cross-pollinated them and he did something which you call as reciprocal cross-pollination. Now, what is reciprocal cross-pollination? What he did was pollen from this tall plant was dusted on the dwarf flowers and pollen from the dwarf flowers were dusted on the tall plants. Okay, so this is what we are calling as reciprocal cross-pollination. But in his case, the results, from this, the one that we are going to do now, or from the other one, just the reciprocal one, were exactly the same. Okay, so that helped him to give forward the different proposals that he had given, which we call as Mendel's theory. There were 4 theories which were proposed by him initially, but later it was found that some examples are present which does not comply with these theories that he had given us. So 2 theories are not considered anymore, they are discarded. Only 2 theories or postulates, as we call, of Mendel are now accepted, which we will do later. Okay, now let us see what happens from these when we are dusting the dwarf flowers with the pollen from the tall flowers. So I will just write this as P1, okay, as tall, and this is P2, this is dwarf. We cross them when we mean there are 2 different individuals which are completely different from each other, that is the characters that we have chosen are contrasting at least, traits. Here we are using this term trait. Of course, the character has to be just one. We are talking about the height of the plant, but here we are talking about 2 contrasting features or traits of the flower, the plant height. Now, as these he had termed as P1 and P2, similarly, the successive generations or the filial generations, as we call filial, he had called as F, the filial generation. So the first generation, second generation, third generations, like that it will come. And, consequently, it will be like F1, F2, F3, and so on. Now, these is what we are calling a simple cross, because when we come next, we will come to different terms like selfing and all these things. So this is just a simple cross when 2 different individuals, okay, at least differing in one character. okay, is chosen, then we will have like these. What he saw, what Mendel saw first of all, that in the first filial generation or F1 that he had called, all the plants were tall in nature. All the plants were tall. That means 100% of these plants were tall in nature. Similarly, if you remember about the reciprocal cross that we have spoken of, the tall plants being dusted by the dwarf pollen, they also produced only tall plants in successive generation. We are talking about the first generation only the F1. Okay, results from each of the character that he had chosen, the 7 characters that he had chosen. The first one is okay. We will just call it as the height of the flower. Sorry, the height of the plant. Next is the color of the flower, the seed coat color, the texture of the seed coat, the position of the flower, okay, then the the structure of the what do you call this pod, okay, the color of the pod. Okay, pod can be inflated or constricted. The color of the pod can be gray, green, or yellow, just like the color of the seed. And the flowers can be terminal or it can be axial. Okay, in each case, what happened was one of the traits, okay, did not show in the first filial generation. Only one of the traits were persistent throughout all the characters he had chosen. You understand what I mean? If we are just showing tall and dwarf here, and we are telling it is the tall character which was actually expressed in 100% of the next generation pea plants. Okay, this will be the similar thing if we are choosing the color of the flower as red, and the successive generation in F1, that is in each of these 100% of these will be red in color. Okay, or the seed coat color will be yellow, or the pod color will be yellow, or the pod will be inflated, like that. Only one of these 2 traits we have chosen, but only one of these will be actually expressed in the first generation. But does it mean that the other trait that we had taken is actually disappearing from the generation? You understand what I mean? What we're telling is, in any case, whatever we are taking, red or white or inflated or constricted, like that, tall and dwarf, only one character is expressing in the first filial generation, the other is not appearing here at all. So the question is the other trait? I'm not using the term character now, I'm using the term trait. Okay, is the other trait completely disappearing from the population? That is not so, because Mendel had taken 2 tall plants from the same generation. Okay, F1 generation, 2 tall plants were chosen by Mendel, and because he is choosing 2 individuals from the same generation, expecting them to come or almost equal. Uh, okay, now I can tell you that equal kind of genes which are present in them, but that was not in his case. He just had taken 2 tall plants thinking that they will have something similar in them and crossed them. Okay, but this is not what is a normal cross. In normal cross, we will choose 2 completely different individuals which do not have any similarities with each other for at least 5 to 6 generations. And now this similarity which has been continued for 5-6 generation is what we call as true breeding individuals. We have used this term before, true breeding. I told you, but this is what is true breeding. That means if some plants are tall in nature, they had been tall for at least 5 to 6 generations, like that, before that. And if you're talking about true breeding dwarf plant, this means the same thing too. It means at least for 5-6 successive generations, they had been dwarf in nature. Is that okay? So if it is like that, then you can understand the 2 parents that were chosen are not at all similar to each other, completely different. But here, in this case. So we are calling this as a simple cross. But when we are taking two tall plants from the same generation, because their parents had been similar, we cannot use the term cross here. Okay, simple cross we cannot take. We will instead take another term, I'm sorry, yes, okay, self-crossing, or in short, we can just call this as selfing. I hope this is clear to you. Cross term or crossing is the term when we are choosing. We have to be sure that the parents that we have chosen from them are not similar to each other at all. At least for 5 to 6 generations, they should be breeding through. And if we are using the self-crossing or selfing, then we mean the parents of them are the same. Okay, I hope that is clear to you. So that is selfing. When the selfing between two tall plants were done by Mendel in F2 or second filial generation, what he found was 75% of the plants were tall in nature, but at least 25% of the plants were dwarf in nature. That means this dwarf trait that we did not find in F1 generation at all did not disappear. We can just say that this simply did not appear in the generation for some reason. We have to find out the reason now. I hope till this it is absolutely clear to you. What we are telling is in F1 generation, we are getting 100% of the individuals tall in nature, which is one of the parents' character or other trait. And what we are telling, the other trait did not appear in the generation at all. But in the other generation, what is happening in the next generation, the successive generation, what is happening? Yes, one of the characters that we are talking about appeared in most of the individuals. So we will say 75% of the individuals were having this trait, and the other 25%, okay, inherited the trait which were present in the first P2, okay, or first P2 or P2, whatever you say. Now, this is up to you, whichever you choose as P1 or P2. But there is just a usual thing that is taken. We say that P1 is the one from where we take the pollen usually, so that is chosen as the male. Or you can say the trait of which is expressed in F1 generation is usually called as P1. Okay. So as you can see, the tall trait has appeared in 100% of these individuals in F1 generation. So we have chosen to call tall as P1 or the first parent, and the dwarf one, which did not appear in F1 at all, but has appeared only in a small percentage, 1/3, okay? Or rather, not 1/3, that is 3/4 of the population in this is what we are calling as the P2. Now, this whole cross was actually given a name by Mendel. Since he had taken only one character for this, which was the height of the plant, he called this kind of crosses as monohybrid cross. Okay, here we come to the terms we had spoken about before. The first one is monohybrid cross. Monohybrid simply means we are choosing one character at a time, but when we are talking about one character, we have to remember that we are choosing two contrasting traits or features of the same character. Okay? Now, we can see that only one of the traits had appeared 100% in its full measure in the first generation, and it is not allowing the expression of the other feature or the other trait that was present in the parent. Okay, or in other words, it is simply not allowing the expression of the dwarf feature tall. If you are taking tall feature here, then in F1 generation, the tall feature is just not allowing the expression of the dwarf feature. Okay, so this is what we are calling as a dominant feature. You all know what is dominant. Someone or something which does not allow the expression of the other when in presence. Okay, so we are calling this as dominant, and the other one, the other trait, which did not appear in the first one generation, okay, or first filial generation, we will call this, because of the presence of the other trait, is something which we are calling as recessive. Hope this is clear. It simply means one of the traits which is going to express in full in first one generation, not allowing the expression of the other trait, is what we are calling as dominant. And the other one which does not express itself in the presence of the dominant one is something what we are calling as recessive. Okay, so we have come to know of many terms here. First was true breeding. Then we have seen what are parents, P1 and P2. Then we have seen what are filial generations, F1, F2, F3, like that, subsequent ones. We have seen crossing, simple cross, and we have also seen self-crossing or selfing. In short, we will just mention this as selfing. Okay. Now, there is a ratio which has been given by Mendel according to the expression of these. In F2 generation, what happens, we have seen that tallness, of course, this is going to dominate because maximum of the individuals will have this trait, but at least 25 of the other trait will be expressing itself. Okay, so if I'm taking 100 individual, I'm telling 75 of them is having tall feature and 25 of them having dwarf feature, or 75 is to 24, 25. Now, if you take the whole thing as 4, this is coming in 3 and the other one will be 1, right? So this is what we are going to take in case of the ratio that was given by Mendel. Since this is a monohybrid cross, the ratio will be known as monohybrid ratio. But here again, okay, we will talk about it later in the next class because I don't think we will have enough time to explain more in today's class. So ratio here will be 3:1. It's simply that the dominant feature is going to express at least 75% are tall, and the other one here will be dwarf in nature. 3:1 ratio. We call this as monohybrid ratio, but is there anything? If there is a problem, you will please tell me. Ratio is 3:1. And here we are going to use a term. Monohybrid we are talking about. Monohybrid we have already said that it is the monohybrid ratio. But what we have chosen here is just the appearance, the physical appearance of these plants, which we can see, tall or dwarf or anything that is physically visible. Okay, that appearance, the physical appearance or the visible appearance of any individual is known as the, anybody can write this? I have seen you all messaging. This is known as phenotypic. Any visible structure or character, okay, is known as phenotype. Phenotype, so from phenotype, we have this phenotypic, this 3:1 ratio of the monohybrid cross that we have just seen is known as monohybrid phenotypic ratio, or just monophenotypic ratio, which we are calling as 3:1. I hope this is clear to most of you. The time is almost over. So I will give you something to do at home. Please understand, genetics is something, if you don't practice it at home, this is simply not possible. If you just attend classes and think that you've understood this is simply not going to come to you. Okay. So you can take from the 7 characters that we have spoken about from Mendel, you can take any other one. And as we have spoken about this dominant and recessive, in the 7 characters, the other 6 characters, that is, there will be such kind of dominant and recessive feature. If you're talking about the character height of the plant, we have already seen this. So that was our first character, even though for Mendel, that was not the first character. This is just for explanation. Mendel used these. We also use this as the first character. You can choose anyone. Say color of the flower. There will be one character. Again, you have to remember this. We are talking about one character, flower color. But there will be 2 traits of it, or 2 variations, or 2 contrasting features that we are going to consider. One is red and white. Likewise, terminal, axial, okay? Uh, then inflated, constricted, green and yellow, or rather, dominant one will be yellow, and the the green one will be the recessive one. So you can choose any of these and you make such kind of phenotypic cross. You can send me, or you can just show it. Next class, you can just raise it and you can show me these characters so that I can understand that you have understood anything. Everything that we have spoken about in this class. In case something is not clear to you, please inform. If I don't get feedback from you, then it will not be possible for me to, help you to understand the subject. So with this, I'd like to conclude today. Thank you very much, everyone. Bye.